As I prepare my sample for a clarification step, I thought I'd clarify what people mean when they say clarification. Basically, a clarification step in terms of biochemistry is when we take a solution that's kind of like cloudy, there's precipitate, non-dissolved stuff, and we make it clear by like clearing out all that stuff that's not dissolved. And so there are different ways to do this, and different industries have different things what they might mean by clarification, but often we're doing some sort of centrifugation. So we're spinning the samples really fast, and the stuff that's not dissolved is then going to get pulled to the bottom. It's going to pellet out, and now you're left with this clear supernatant. And so basically the supernatant is going to be the liquid that's left over. So typically we're doing this with some sort of lysate. So say you've broken cells open and now you have all that cellular gunk, like the membrane bits and things like this. Now, you're often wanting to do something like purify out a specific protein from that mixture, or do some other step where you don't want all that gunk getting in the way. So we're going to go through all these fancy steps to purify out the proteins and the DNA and the RNA and whatever we want, but we don't want to basically have all this other stuff getting in the way that we could easily get rid of. And so it's easy to get rid of this big gunky stuff with just like a simple centrifugation. And so a quick clarification step is a way to do this. Basically pull out all that gunky stuff that would get in the way if you were to try to do something like even just run an SDS page. And so this clarification comes in handy with lysates. Another time it comes in to play is when you're doing something like a Miniprep. So when you're like selectively precipitating out various things. So in a Miniprep, basically what you're trying to do is you're trying to isolate a plasmid, so a circular piece of DNA, something that you clone maybe that you put into bacteria. And you have to break open the cells and you want to isolate that plasmid. And so you want to precipitate out all the other stuff. And so you add like base and acid and stuff like this, denature all the proteins, get those to come out a solution. You've got the membrane bits, you've got all this gunk. And you're gonna use a column like this that's going to bind the DNA on this membrane and let other stuff through. Now, if you were to take that big gunky lysate and just put it on the column, well, the column would get like clogged, but you'd have all that gunk clinging to the column as well as your DNA, and that's not what we want. We need to clarify the solution before we get our DNA to bind. And so when you're doing a Miniprep, before you put it on the column, you do a clarification step where basically you take that mixture after you've lysed it, and then you take the liquid, you spin it down, you take the liquid part, the supernatant, and that is what you apply to the column. So there's another place you'll see clarification. And speaking of some clarifications, this example gives me the opportunity to say, like, okay, when you clear and colorless are different things. One of the first times I did a Miniprep, it was with the like lice blue. It's like this pH indicator, and so when you add the base, it should turn blue, and then when you add the acid to neutralize it, it should turn colorless. And so, but you're still gonna have all this like cloudy, clumpy gunk in there. And so I was like waiting. I'm like, why is this not turning colorless? It's still white. And I was like, oh, it's colorless, not clear. So that wasn't a great example because thanks to all of that stuff suspended in the liquid, not in the solution, but just kind of like floating around in there, it's gonna look kind of white. The gunky stuff's kind of white. But the liquid part should be colorless, or at least it definitely should not be blue. It might be kind of yellowish because when you have a bunch of proteins, things tend to look yellowish. But so you can have color that is also going to be clear. And that's another way you can get tripped up, is if you have a colored solution that is clear, and then you're waiting for your protocol to your solution, you're wondering why it's not turning clear, but it is clear, it's just a color. So clear just means like we can see through it. There's actually different like types of being able to see through things as well. There's like translucent, which is where it's like kind of cloudy, and then there's transparent where you can see like clear through. But basically colorless just means that there's not color, and clear means that you can see through it. And so we can see through this liquid after we clarify it. And commonly we're doing this clarification with just like centrifugation to spin out all that heavy stuff, and then you're left with that nice clear liquid, the supernatant. And depending on how like solid the pellet is, you might either like decant it, so pour off that supernatant, or pipette it out, so like aspirate it. And When you do this, when you're doing a clarification by centrifugation, you want to work as quickly as possible once you've got that pellet pelleted, so that things aren't like the stuff that pelleted out doesn't try to come back into solution. So for example, if you're doing it in the centrifuge, you want to keep all the tubes in there. Like when you take one tube out at a time, remove the supernatant, then take the other tube, try to like handle them as little as possible so you're not getting stuff to redissolve back into that liquid that you just tried to pull everything out of, and that would be like defeating the purpose of doing it. So work quickly, and be careful, especially if it's like a loose pellet, that you're not pouring some of that pellet back in. The nice thing about clarification is if it kind of resuspends, just spin it again, and yeah, then you can do whatever you want to do. Yeah, so some other places, some other kinds of clarification involve things like filtration. Different industries, like I said, might have different, like if you're working in like big batches and stuff, there might be more fancy ways of doing things. But typically, when just in our day-to-day lab work, we're doing these clarifications with some sort of centrifugation step. So hope that helped. I know it's kind of weird little post, but basically clarification is one of those terms that we kind of just use, and I don't know if anyone actually explains it to people. I didn't really get it explained to me. It's just kind of like a term that was used. I was like, okay. And then when you try to Google it, you get like other definitions of the term clarification, which is not very clarifying. So hope this was.